Hello class, today we will study two phylum which are the Deuteromycota and Basidiomycota. From the phylum Deuteromycota, we will review the pathogens Colletotrichum, Fusarium, and Cytospora and Phoma. And from the phylum Basidiomycota, we will study few orders. Among them, we will review the Ustilaginalis, Uredinalis, Agaricales, and Aphilosporales. And inside those orders, we will review a few pathogens and some diseases, the most important which are smuts and rusts. Our first pathogen is Colletotrichum, and it is the causal agent of anthracnosis in different hosts. The main symptoms that this pathogen can cause are dark spots, sunken lesion with a slightly raised rim, and whenever it infects fruits at young stages, it causes its abortion, so they drop from the trees, and in later stages, they rot. In the slide, we can see different photographs of anthracnosis in avocado. Here, we have how it affects flowers and developing fruits, so they mummify and fall from the tree. In mature avocados, it causes these circular lesions, and same on the tree leaves. The typical symptoms on strawberry are these sunken lesions, the same that we can see in bean, and on tomato. They're usually very circular lesions which are sunken with this upper rim. The asexual fruiting body of this pathogen are the cerbuli, which are setae, as we can see in this photograph. This is a photograph of an agar plate, and here we can see these Haiti-like fruiting structures, which are the cerbuli. Having a close-up of them, this is the way they look, and even at higher magnification, this is how a setosa cerbulus looks like. This is a photograph of a cross-section of Colletotrichum in avocado. So here we can see how the cerbuli form, and we can see different conidia. Typically, conidia from Colletotrichum have this darker center, which is the nucleus. Now we will review one of the most devastating diseases across the world, which is Fusarium. Fusarium can cause different diseases, and among them we have the vascular wilt of different plants. Usually the disease starts with the loss of turgidity of leaves, which then become yellowish, the plant wilts, and then it dies. The most common species causing vascular wilt is Fusarium oxysporum. When we do a cross-section of a plant which is infected with the disease, we usually see a vascular discoloration because the pathogen grows through the vascular tissue of the plant. In this slide, we have typical symptoms of soybean, tomato, and bananas. In the upper side, we can see the typical wilting on the plant, and in the lower part, we can see the typical vascular discoloration of the vascular tissue. Another disease which can be caused by Fusarium is Fusarium head-blight of cereals, and it affects wheat and other cereals, and is caused by different pathogens including Fusarium graminearum, Fusarium culmorum, Fusarium avicennarum, and Fusarium poa. Usually infected spikelets get water-soaked appearance and a straw color, and they also develop a pinkish, purplish color, as we can see in this photograph, which is due to the development of mycelium and conidium in the infected spikelets, and also by the purplish perithecia that the pathogen develops. Infected kernels are usually shriveled and discolored with white-pink, or they have a light brown scaly appearance, as we can see in this photograph. This disease is also important because the pathogens produce different mycotoxins which are harmful for humans and animals. Fusarium has different reproductive structures, and its sexual fruiting body are Disporodokia, which are clusters of conidiophores. This is one Sporodocium right here, and it has kind of a hand shape. And the asexual reproductive structures are also the macroconidia, which are these large structures, which are usually normally the 3 to 5 cells, and microconidia, which are these ones over here, which usually have only 1 or 2 cells, and it also has an overwintering stage, which is the chlamydiospore, which is a thick-walled sexual resin spore, and they are very round-shaped and they have very thick cell wall, as we can see in this photograph. Continue with the Deuteromycota phylum. We have the pathogen Cytospora, which causes cytospora canker, and different woody plants. This pathogen produces oranciri, which are tendril-like masses of exudate spores, which are held together by a slimy matrix, as we can see in this photograph. Now we will review the pathogen phoma, which causes phoma blight in different vegetables and annual plants. Here we have a photograph of phoma in tomato on the leaves, which cause these circular lesions, and on the fruit. The sexual fruiting body of phoma are the pycnidia, which are flask-shaped structures which contain the conidia. Here we have the pycnidium, which is kind of a flask, and on top of it we have the ostiole, which is the opening, natural opening, where the release through which the spores are released. And here we have a photograph with the stereoscope of a culture in an agar plate of phoma. This is how the pycnidia look like in the petri dish. Now we will review the Basidiomycota, and to start with this phylum, we will review the order Ustilagenalis, which are the different pathogens which causes the smooth. We have at first the pathogen Ustilagonura, which causes loose smooth in barley, and Ustilagothritis, which causes loose smooth in wheat. Usually infected plants do not exhibit any symptoms until the head in development, and as we can see in this photograph, the heads are replaced with teliospores. That's why we have this brown color in the wheat spikelets. And usually during the flowering, infected plants look taller and they develop faster. That's why they show a higher phenological stage compared to all the other plants. Another pathogen from the order Ustilagenalis is Ustilagordae, and it causes the disease covered smooth of barley. And in this case, the grains are replaced by dark brown spores, as we can see in this photograph over here. And usually the infected heads are hard and compact. Infected plants look shorter, and these smooth masses do not break up readily as in the loose smooth that we reviewed before, because the spores are held tightly. And now we have stinky smut, or common band of wheat, which is caused by Tiletia tritici and Tiletia laevis. And in this pathogen causes the development of shorter plants, and the infected kernels develop into bond balls, as we can see here. And the seed coat remains intact. this is how infected plants look like. Here we have healthy grains, and here we have diseased grains. And there's also the stem moth, which is caused by Ustilago hypothetis, which only affects grasses, and it produces brown sorry on the infected stems, which causes them to become black during the seed filling. Infected stems occasionally flower, but they do not set seeds, and the plants are usually shorter. A very interesting pathogen from the phylum Basidiomycota of the order Ustilaginalis is Ustilagomatis, which causes the corn moth, and it affects any part of the plant, causes the development of tumor-like galls that contain most of Suriteliospores. An interesting thing about the corn moth is that in Mexico they usually eat disinfected plants, and that's a very typical traditional food for Mexicans. Now moving to Uriadinalis, which are the pathogens that causes the rust. Different rusts. We have at first the Endocrinatum harknessii, which causes the western gall rust. And when this pathogen infects the plant, the cells grow and divide quickly, and it starts the formation of a gall, which takes between 2 and 4 years. And usually the galls are covered with yellow to orange ichthyospores, as we can see in this photograph. Another rust is Puccinia graminis form special tritici, which causes the stem rust of wheat. In this pathogen, different oval structures develop in leaf and stems, and they have uredospores, which are these kind of orangey and powdery structure. So when you take them, when you just pass your finger through them, you're going to have lots of uredinospores in your finger. Here we have the uredial pustules. Inside we have the uredospores, and when the uredospores become visible, it is because the epidermis of the plant ruptures. We have healthy tissue over here, and usually around the pustules, we have a chlorothic area surrounding them. This pathogen, in order to complete its life cycle, needs 2 different hosts. The first one is the wheat, and the second one is the barberry. So barberry is considered the alternate host for Puccinia graminis from special tritici, and infected leaves of barberry become hypertrophied, as we can see over here in the photograph. And in the upper surface of the leaf, spermagonia or pycnia develop, while in the lower surface of the leaf, the aesia develop. The aesiospore in the aesia are the ones that spread through wind to it, and they germinate and continue the life cycle of the pathogen in wheat. Now we will review the order Agaricales, and here we have the basic structure of any mushroom. So here we have the cap, which is the upper surface. The filaments-like structures are the gills. And here we have the ring of the mushroom, the stalk, and here we have the mycelial connection. On a cross-section of a fungus, we have the same parts that we reviewed before, the cap, the gills, and the stalk. And when we have a close-up of the stalk, we can see the trauma, which is this region, which joins the hymenium, the subhymenium in both sides. So hymenium, subhymenium, hymenium, and here we have the basidiospores that develop in a basidium, and they're kept together by an esterigma. This is another cross section, but it is an horizontal cross section. So here we have the trauma, subhymenium, hymenium, which is where all the basidiospores develop, basidium, which is where the basidiospores grow and develop, and the esterigma, which are the structures which keep together the basidiospore to the basidium. Now we will check the order Aphylosporales, and here we have one important pathogen, which is Enterobasidium, which causes stem rot in different perennial trees. This is one of the most important forest pathogens, and here we have the fungal conch, which is the fruiting body of the pathogen, and here the basidiocarp develops. The lower surface of this conch is usually made of different pores where the basidiospores develop. The sexual spores are the basidiospores in this fungus. Here we have a cross-section of Polyporus, and here we have the pores that develop in this conch, and here we have the spores in a close-up. Another pathogen is Phelinus tremulae, which is the causal agent of white hairwood of rot of aspen. Here we have the conch, how it looks like, and how it affects the wood in the tree. Another pathogen is Phelinus spini, which causes the white pocket rot, and this pathogen develops perennial cocks, and in the undersurface of young cock, undersurface we have the young conch, which is usually yellowish brown, and it has large and irregular pores. These white pockets develop where the conchs develop, and here we have very small spindle-shaped white pockets, which are caused by the degradation of lignin. So this is everything for today. Go and continue with your lab quiz, which is on eClass, and using all the information on the slides and the video and the lab guide, answer the questions. You will be able to complete the lab quiz until 1 hour before the next lab session, and don't forget that you can go back and forth as many times as you want, and once you complete the lab report, don't forget to click on submit. Enjoy your lab. Have a good day. Bye.